Okay, so welcome to this next video in which we are discussing the adaptive immune response. Okay, so we had just discussed that when the antigen-presenting cell, this professional antigen-presenting cell, which is either a dendritic cell or a macrophage, comes into the lymph node, and so far we've just drawn the one which has an antigen fragment presented on MHC class 2. Then basically what it will do is it will percolate through the cortex and the paracortex of this lymph node, looking for a T cell, a CD4 positive T cell, which has a complementary TCR, T cell receptor, on its surface for this antigen fragment that is presented on the MHC class 2. Okay, then what will happen is the antigen fragment in the MHC class 2 will bind to this T cell receptor on the surface of this CD4 positive T cell, and the CD4 will then bind to the MHC2, and that will deliver signal 1 to the CD4 positive T cell. Okay, then that's not enough to activate this T cell. Instead, what you need is co-stimulatory molecules expressed on the surface of the professional antigen-presenting cell, and the expression of these co-stimulatory molecules will be induced by the activation of pattern recognition receptors within the APC, which will be activated by pathogen-associated molecular patterns. So this is a checkpoint, basically, to make sure that this antigen fragment is actually from an invading pathogen. And the co-stimulatory molecules you have are CD40, which binds to CD40 ligand on the surface of the T cell, and B7.1 or B7.2, which bind to CD28 on the surface of the T cell. Okay, this then leads to signal 1 and signal 2. Well, this half here is going to lead to signal 2, and when you've got signal 1 and signal 2, they're going to cause something to happen within this T cell. Okay, so what will happen is the T cell will start creating interleukin 2. Okay, so it will start secreting interleukin 2, IL-2. Okay, and why is it going to do this? Well, basically, it is going to take part in an autocrine stimulation loop. So what it also does is it synthesizes the final piece of the interleukin 2 receptor. So it starts producing also the interleukin 2 receptor alpha piece. So this stands for interleukin 2 receptor alpha piece. So let me just write this out. So IL-2 stands for interleukin 2. Okay, and this is the interleukin 2 receptor alpha. Okay, so on the surface of the T cell, you will always have the beta and the gamma components of the interleukin 2 receptor. Okay, so let me show this. So if this is a piece of membrane of the T cell, then you will always have the beta and the gamma portion. However, that is not the complete receptor. So let's say this is beta and this is gamma. To make the complete interleukin 2 receptor, you need to produce the alpha component. So once the T cell has received signal 1 and signal 2, it will start producing the alpha component here. Okay, and that will make the complete interleukin 2 receptor here. So this is the interleukin 2 receptor. The activated T cell will now also produce interleukin 2 and secrete it out from the cell, and then that interleukin 2 will come back and stimulate the receptor on the same cell, basically. And this kind of a signaling pathway is what's known as autocrine signaling. Okay, auto means self, basically. So this is a signal on yourself. Okay, so autocrine signaling. Okay, and then what's going to happen is that the interleukin 2 receptor will lead to signal 3 going to the T cell, and then things start getting interesting. This will trigger the T cell to differentiate and then start dividing, which we'll discuss more in a moment. But for now, what I want to do is temporarily go back and talk about the CD8 positive T cells. So we talked completely about the CD4 positive T cells and abandoned the CD8 positive T cells. So what if our antigen presenting cell comes with a fragment attached to MHC class 1? Okay, so let's draw this out. Well, thankfully the entire process is almost identical to that for MHC class 2, just on these different types of T cells. So remember, in the cortex and the paracortex of the lymph node, you have loads and loads of T cells, and some of these will be CD4 positive T cells, and some will be CD8 positive T cells. Okay, and within those populations of CD4 positive and CD8 positive T cells, you have a huge amount of variation because the T cell receptor won't be the same between any two T cells that you pick. It will differ in this antigen fragment binding region. Okay, so the starting point is that you're going to get this antigen presenting cell here coming in with its antigen fragment presented on major histocompatibility complex class 1, MHC class 1. Okay, and remember, this is only if you've got an intracellular pathogen. Okay, so if you've got an extracellular pathogen, you'll have had to phagocytose that pathogen, and its antigen fragment will be on MHC class 2, which is where you present an antigen fragment that you got from a phagocytosed pathogen. Okay. However, you will also be able to phagocytose intracellular pathogens whilst they're in the extracellular fluid, and therefore intracellular pathogens will generally also trigger this sort of a response, which will be important later. Okay, so now what we're talking about, however, is purely intracellular. This applies only for the intracellular pathogens. So if you've got an intracellular pathogen, you will put its antigen fragments on MHC class 1 proteins here. So let's colour in this MHC class 1 in vivid purple. Okay, here. And we've then got this antigen fragment mounted in the cleft of the MHC class 1. Right, so the antigen presenting cell has searched long and hard for a T cell that has a T cell receptor which is complementary to this antigen fragment. So here is the T cell receptor bound to the antigen fragment. And now I'll draw CD8 on this side, and there's no significance to that, don't worry. It's just because I want to leave room for the co-stimulatory molecules. So here is CD8, cluster of differentiation 8. Right, so if you've got an antigen fragment which you want to present to a T cell receptor, that is on the MHC class 1, you must present it to a T cell that has CD8, because the CD8 must bind to the MHC class 1 in order for this to deliver signal 1 to the T cell. Okay, so here's CD8 in blue, and here is the T cell receptor in orange. Okay, right, so the binding of the T cell receptor to the antigen fragment on the MHC class 1, and also the binding of CD8 to the MHC class 1, is going to deliver signal 1 to this T cell. Okay, so this is going to deliver signal 1. Okay, and I'll put this here. Signal 1. However, again, signal 1 is not powerful enough to actually activate this CD8 positive naive T cell on its own. Basically, you need co-stimulatory molecules to be present on this APC. So again, if this genuinely is the antigen fragment of a pathogen, then basically this pathogen will have a huge number of PAMPs, pathogen-associated molecular patterns, and these will have activated pattern recognition receptors on the surface of the antigen-presenting cell, and that will have caused the activation of the expression of certain co-stimulatory molecules, which I will draw here. So these co-stimulatory molecules are exactly the same, thankfully. So one of them is going to be CD40, here, and the other one is going to be either B7.1 or B7.2. So I'll write B7.1 /B7.2, and that means either B7.1 or B7.2. It doesn't mean some sort of chimera of both of them. Okay, so here is B7.1 or B7.2 in pink there. Then in red we'll have CD40 as the other co-stimulatory molecule, and again these bind to the same receptors on the surface of the T cell. Okay, so we have CD40 ligand here, which binds to CD40, and then we have CD28, cluster of differentiation 28, which binds to B7.1 / B7.2. Okay, so let me put these in. So here is CD28 in turquoise. Okay, and here is CD40 ligand, which we'll draw in blue again, because I'm running out of colours. And I can't bring myself to use yellow, it will never show up in this light. Right, so when CD40 binds to CD40 ligand and B7.1 or B7.2 binds to CD28, this delivers signal 2 to the T cell. And then what will happen is, again, this T cell will produce the interleukin 2 receptor alpha portion, which will then combine with the already expressed beta gamma portions of the receptor, and then the T cell will also produce interleukin 2, which will then go around, stimulate the interleukin 2 receptor, and that will deliver signal 3 to the T cell. So the process of T cell activation is almost identical within naive CD4 positive T cells as it is in naive CD8 positive T cells. Now, the processes now diverge basically. So what we're going to begin with is we're going to abandon CD8 positive T cells for the time being. We'll come back to them later, and then we're going to concentrate on CD4 positive T cells, and specifically we're first going to look at humoral immunity, because remember, CD4 positive T cells will be activated by both intracellular pathogens and extracellular pathogens, because this was the one that was activated by both. It was mainly extracellular pathogens, but remember, when the intracellular pathogens are extracellular for a short while, they may be phagocytosed, and their antigens will then be processed and presented on the surface of MHC class II molecules. Okay, so they will activate CD4 positive T cells. So CD4 positive T cells also have something to do with the the cell-mediated immune response, but we'll initially concentrate on the humoral response, then we'll come on to the cell-mediated response, and we'll return back to the CD4 positive T cells, the CD8 positive T cells then. Okay, so see you in the next video.